Greetings, everyone, and thank you for joining us for the GROW webinar. It's my pleasure to introduce our speakers today, Emily Coffey, Jean Linsky, and Dan Carver. The speakers will present information on the emerging gap analysis and metacollection management tool called Gamma, and will provide a live demo of the tool at the end of the presentations. Dr. Emily Coffey is Vice President of Conservation and Research at the Atlanta Botanical Garden, where she leads a team of conservation scientists and horticulturalists advancing rare plant propagation and ecosystem conservation across the southern eastern U.S., the Caribbean, and the Americas. Jean Linsky is the coordinator of the Global Conservation Consortium for Magnolia. In this role, she collaborates with botanic gardens worldwide to develop and implement conservation strategies for threatened magnolia species. Dan Carver is a spatial scientist specializing in developing open source decision support tools and end-to-end data pipelines. As an expert in R programming and Linux environment management, he has led the technical architecture of geospatial initiatives while maintaining a foundation in applied geographical research and plant conservation. The title of their presentation today is Developing a User-Friendly Gap Analysis Tool to Build More Complete Situ Collections. Welcome, Emily, Jean, and Dan. Thank you very much.
Thank you so much for having us today. So this is really exciting for us to be able to talk to you today about the new tool Gamma. And I'm going to present a little bit at first on why we actually came to develop this tool. And this is a tool that I'll give you a bit of background. Jean will give you an example of how this tool is being used and some of the outputs of the tool. And then Dan's going to walk you through an actual tutorial, so you'll get to actually see what the tool looks like in real life. So a huge thanks to both of my co-presenters today, and we'll get started. So this work is done in collaboration with Botanic Gardens Conservation International. And the mission of BGCI is to mobilize botanic gardens and engage partners in securing plant diversity for the well-being of people and the planet. It's a very extensive network of more than 3,500 botanic gardens worldwide. You can see here an image of all of the different gardens that are represented across the world. And botanic gardens are really important because of the number of visitors that we get, as well as the botanical expertise that we host in these gardens. And part of what BGCI's mission is really to focus on the conservation components of a botanic garden. And they developed this integrated plant conservation concept that really focuses on ways in which the in situ work, or the work that we're doing in the wild, is deeply integrated into the ex situ plant conservation that's occurring in the gardens themselves. And you can see here that it's everything that from our restoration work, our collections work, and how this is all sort of connected to the research, horticulture, and education components of the garden. One thing that's really important, you'll notice the purple boxes. This is the work that we're focusing on in our talk today, and where our tool really focused the work on. So this is our ex situ work. This is all of the material that we're collecting out of the wild in the gardens. And we have everything from our traditional germplasm material, our living, which includes our seed banks, our cryo, our micropropagation. And then, of course, our living plants, which are our conservation collections, reference collections, and even our display plants. And we are actually adding this additional box, which is our meta collections box. And this is where we, as a team, that I'll introduce all of the individuals that are working on this project, we're really excited to add more and contribute to the work around meta collections, which, quite simply put, is a collections of collections that are collectively managed. And so, to set the scene about sort of why we are here and the work that's being done through a very strong collective called the Global Conservation Consortia, I wanted to give you a little bit of background information. So back in 1999, the Global Trees Campaign was established, and it was a joint initiative between Flora and Fauna International and BGCI Global. And this work was really focused around cataloging the world's trees, prioritizing those trees and the conservation actions that it would take to secure those trees from going extinct, and then trying to really take direct conservation action with local partners, all focused around the zero tree extinction hope. And then out of that has come several additional initiatives through that Global Trees campaign. And a really pivotal piece of work that came out quite recently in the last few years is the state of the world's trees. Through the work of the Global Tree Assessment, the concept around establishing consortia, these global conservation consortia, focused around tree species to begin with, was really evolved and developed. And we started working on this concept of these global conservation consortia based off of the knowing of sort of how threatened so many of our tree species were, and that we needed to actually mobilize a really strong coordinated network of experts so that we could collaboratively develop and implement actual conservation activities. So this was less about sort of the planning and really about the action. We knew with the state of the world's trees that 1 in 3 species are threatened with extinction. We knew that we really needed to scale up conservation action on the ground, and we wanted to create something that would focus not only on the ex situ work, but also in situ work in centers of diversity. And so that is where BGCI initiated this GCC concept, or Global Conservation Consortia concept. The GCCs have at the beginning really focused on tree groups, like I was mentioning, but we have actually shifted to be more inclusive, which is quite exciting. And it's really based off of several key criteria. So the criteria that we utilize to help us evaluate what would make a good consortia are things like whether they are a highly threatened group, whether they actually have difficulty with challenges growing. So things around, orthodox versus recalcitrant. So we have been focusing on species that are more recalcitrant. We're also looking at species that are quite challenging to manage. So things that have never been grown in collections before, that people have very little experience with. And of course, we're looking at groups of plant species that have sort of low capacity in their diversity centers, and that there's an ability and excitement around building that capacity in those centers of diversity. We're also really interested in species that span across many regions, that allow for greater utilization of a coordinator and someone who can actually bring people together. And so many of these key criteria that we have utilized have been very important for us developing what is the current slate of conservation consortia. So this is the current group of consortia, and you can see very excitingly that we have more than just trees now. And I think a very exciting note to this group is the food plants group. I, in partnership with Jean, lead the Magnolia GCC. And both the magnolias, the oaks, and the cycads are some of the older groups that have been around since around 2018 is when a lot of those groups were starting to form, in 2019. And so we've actually had several new groups come on, and we have a lot of working action around all of these different groups. So we have a set of major objectives that all of our consortia are forming around. And while each consortia might do some of these objectives slightly differently, we actually focus as sort of our primary work around these centralized concepts. This is to give us a framework to work together, since we all have different specializations within our vast different taxonomic groups. So the major objective is really fostering this network and bringing people together so that we can have really strong activities that we're all focused around. We also do a lot of work around identifying and prioritizing species of greatest conservation concern. And so the tool that we're utilizing will help us with that priority. And then we also are really interested in developing very strong, high conservation value ex situ collections. This is where we're really focusing around the genetic diversity of the collection and trying to ensure that we have both temporal, so through time, and spatial, that geographic distribution of our ex situ collections, so that if something happens to a collection or a population in the wild, we actually have really strong genetic diversity backed up in our ex situ collections. We also focus our major GCC objectives around research, anything that can be done in situ to ensure that the populations that are left are still maintained. Additionally, we are focused a lot on building capacity in country and in these diversity centers. So this includes everything from writing grants together to looking for funding, doing trainings and workshops in those diversity centers so that more of the work can actually be focused in these diversity centers. And of course, doing things like building public awareness that we're doing today and trying to engage a broader community around this work. And of course, I've already mentioned this, but raising money. This is really important so that this work can be carried on. And so funding is really critical to the work that we're all doing for the GCCs. This is a sort of a diagram of how structurally these GCCs work. And we focus at Atlanta Botanical Garden, we are the global lead. And this is where we all share sort of this all of the GCCs share this structured format to help provide some clarity around who's doing what and give really strong catalysts to move this action forward. So we focus as the global lead for magnolias, and then we have different representatives, steering committee representatives, represented across our different biodiversity hotspot regions for magnolia. And those steering committee members are really our strong points and contacts in the community that then help us reach out to our species stewards who are helping do a lot of on-the-ground work, and to the community of the network of community members we have individuals joining from NGOs, universities, private landowners, state and federal agencies that are joining and participating. And so it's really about a very collaborative, supportive network that we are all actually focusing together to ensure that we have multiple consortia that can actually work together, as well as people within our consortia being able to really strongly support one another with the work that we're doing and share information, share data, share knowledge, and be able to really have meaningful outputs towards the betterment of these different species we're focused on. So part of, like I had mentioned, those objectives are ex situ collections. And so that's what the remainder of this talk is going to be about, is the work in which we have been focused on this gap analysis work and the kind of data that we're collecting and trying to collect for this sort of work. So gap analyses are not new, and you all are very well aware of this. Understanding gap analyses are really one of the additional tools that we have taken from the crop world, in which at the early days, gap analyses were being performed, and it was a really strong amount of work that this community in the crop world was actually starting the work around these gap analyses and understanding sort of what is missing both geographically and in collections genetically. And so what the conservation community has done is really learn from all of the amazing work that has been this, understanding and the origins in the crop wild world. And so the recent work that we've been focusing on has really occurred through learning from the work that's been done within the crop wild the crop and crop wild realm, and really trying to build off of that for how we could apply that to conservation work. And so these are some really great papers. I'm sure many of you are very aware of them. We're happy to put them in the chat at the end, any links to them. And then these are some of the examples of works in which our field, the conservation field, has been utilizing. So we have gap analyses for conservation that have been occurring in oaks, walnuts, and magnolias. They're occurring at various different scales, from both a sort of regional, so US focus, to global focus, and in some cases, even more specifically, very small regions such as the southeastern United States, for example. And the work was really initiated and started by some incredible partners that are part of this work that we're doing here. So Colin Curry, a huge thanks, Dan Carver, who you'll hear from, and Emily Backman, who was formerly at the Morton Arboretum, have both really focused on this in situ, ex situ work that has been brought into the Botanic Garden world. So what we are doing in our gap analyses is, and within the work that we do already in Botanic Gardens, is we collect a lot of accession level data. We utilize one of the tools called Plant Search, and this is a tool from BGCI, Botanic Gardens Conservation International. And this is where gardens are able to upload their information and data on material that they have in their ex situ collections. This is part of a whole host of tools that BGCI hosts for gardens. And so we're actually leaning in to utilizing this data. So the data that we're focused on for our collections is, of course, ex situ conservation collections that have high value or high genetic diversity. And we're also really interested, of course, that accession level data, so individual tracking. And this allows us to then be able to develop these gap analyses tools that we actually have. So what we can see is that we're really trying to work towards a comprehensive species diversity in our conservation collections. So these gap analyses allow us to really be able to ensure that we do not have a large gap in our wild genetic diversity and what is represented in collections. And we're trying to ensure that we're really closing the gap by ensuring that we don't have any missing genetic diversity in our collection. And the reason that we actually are using these gap analyses is so that we can do a better job at curating our collections. So questions that we're asking, of course, from our collections are: how genetically complete are our collections? These are things that had not necessarily been utilized and a focus of most of our ex situ collections in earlier days. We're really interested in sort of what is the current diversity within the collections and what is not in our collections. Are there any plants that are redundant that we don't need to be maintaining, or that we could be sharing with other gardens for additional backup? And should we be bringing more plants into collections, and sort of how often should we be doing that? So the gap analysis that we were focused around is, of course, as all of you probably are aware, utilizes R and requires a great deal of time and expertise to either learn how to utilize R if you haven't utilized it before, and how to run the code and understand any issues that might be happening. We're often focused around working with botanic garden curators who don't have time to obtain new expertise in this. And so this is one of the reasons that we were so excited about building this tool, and our group came together and said, we need to find a way to actually make these gap analyses really approachable and fast, and something that a curator that doesn't have time to invest in learning R or any coding program could actually utilize. And so that's where the development of GAMMA, or the Gap Analysis and Meta Collection Management tool, came from. So what Gamma uses is both a visual output that is able to quantify and assess the data that is at a session level data that gardens are already tracking for their material. And if they aren't already tracking it, then they can start tracking that to allow them to actually be able to participate in utilizing this tool for their collections management. And so this is really focused around the geographic and ecological coverage and also, of course, the gaps that we're seeing in our collections. And this is a really applicable tool for people to be able to just pick up and utilize with fairly little training. The kinds of data that we have been gathering are the kinds of data that many of our partners in botanic gardens that are doing field collections might already be collecting. So we're trying to make sure that we're not asking really huge new lifts of them. So these Situ collection records are really focused around places where we have information of the material in the collection or in the gene bank that we know where the material came from. And that we also have our wild occurrence data, so where this is occurring from in the wild. So we must have our records of in situ and our wild data collection points to be able to input this. The outputs look like this. You're going to get to see this in just a minute. And this is where we're really able to assess this geographic and ecological gaps and the coverage strongly through a visual representation. We also, with the tool, are automatically calculating the representativeness scores for comparison and prioritization. And also we create an output you can see here. I don't know if you can see my cursor all, but we have these are the various different outputs that you can actually see with our conservation gap scores. You can see the various different areas in which the map is representing. You can see whether or not some of the germplasm is represented. So that's our species observation, where we actually have material and where we don't. And so you can actually see this really strong visual output that allows us to be able to quickly and visibly see what is in collection and what is missing from our collections and what kind of representation we have. And so as if you could see my little cursor, then you could see we have in the bottom corner next to the map is our conservation gap scores. You're probably quite familiar with these sort of scores where we have our various different sampling, geographic, and ecological representation, and whether you can actually, this will be automatically calculated for you. And so this is sort of aggregated in this final table. And this really gives people a very quick output that is really fast. And we have been hearing great feedback from our sampling teams that they're really excited about the visual representation of this and how quickly they can input their data to see what's missing from their collections and where they could focus some of their new collections work. And so before I hand it over to Jean, who will actually go through one of our gamma use cases for Magnolia pyramidata, I just want to acknowledge this incredible team of individuals who have been working on both this concept of meta collections and these collections that are collectively managed for many years. So the work that was started more than 10 years ago around this concept of meta collections. And the current project team, this is everybody that we have working on this project, is currently funded by the Institute of Museum and Library Services and also the U.S. Botanic Garden out of Washington, D.C. And we're so grateful for everybody who puts in countless hours to be able to make this tool really usable and approachable for our curators through Botanic Garden work. So I'm going to hand it over. I'm going to stop sharing and hand it over to Jean to give you an example of some of this, the gap analysis in action.
Great. Thank you, Emily. So yeah, I'm really excited to present an example of how we've used gamma within the GCC for magnolia. And it's a rare and threatened species here in the Southeast US. So as Emily mentioned, as a global conservation consortium, we have this goal of building more comprehensive ex situ collections. And so we think about that in terms of a few phases, really understanding first, we need to understand the current status of the collections of our given species, then understand ways to expand those collections. So often through joint collecting expeditions, going out, filling those gaps that we identify, and then really the collective management amongst multiple institutions for for managing those collections together. And so Gamma really helps us to achieve that first goal of understanding the current status, and then, of course, over time as we're building the collections. So the species I'm going to talk about today is Magnolia pyramidata, or pyramid magnolia. It's largely a coastal plain species here in the southeast US, and it grows on slopes and ravines in mesic hardwood forest. So it gets its common name that from its pyramid-shaped growth habit. And it has these really spatulate-shaped leaves with very distinctive sort of earlobe bases, really nice white large flowers that come out in April, and these cones that mature in August. So the pyramid magnolia ranges from South Carolina along the coastal plain to East Texas. And it really apparently has these core populations in Alabama and Mississippi with dense population or less dense populations in other states. So you can see through the light green counties, these are the more dense sort of populations with less dense in the yellow county populations. Um, there have been in the past some populations in Kentucky, but these are thought to be extirpated. So in recent years, it was assessed as endangered on the IUCN Red List, and it's very heavily fragmented. And there's been some anecdotal evidence of loss of subpopulations. So the GCC for Magnolia, we're concerned about the decline of the species and really understanding the conservation status and what needs and actions might be need to be taken to ensure that the decline didn't continue. So what we were able to do is reach out to understand the status of the species in ex situ collections, and was to do a gap analysis in ex situ and in situ actions globally for magnolias and make recommendations. So in addition to a global level report that we published in 2022, we published a US Canada level report following literature review and emails and surveys of ex situ collections that were holding different species. So here you can see some of the ex situ results for the US native magnolias, and pyramidata on the far right there is actually one of the least represented US native species in ex situ collections. And so we were able to ask at the accession level of whether the plants were of horticultural, unknown, or wild origin, and summarize some of the outputs from that. And so we were able to dive in deeper with those accession level records and actually visualize those collections using Gamma. And so Emily had actually shown you a clip of our results earlier, but this is some of our data that we were able to get in 2022 and really visualize that there are areas across the range that are not in collections. So the purples are where we have collections, but in particular, at that time, noticing that there were no plants in collections from Georgia or South Carolina. And we were really able to use this information to identify areas to go out and do field collecting. And so in 2024, 2025, and in fact, this year right now, we have started to build relationships with landowners where the species exists to go out and do collections, hiking through some of those ravines, finding trees and working with partners to take samples going to find them in the spring, relocating them in August to do seed collecting, and to bring some of those underrepresented areas into collection. And so I was able to do another analysis in 2026, some subtle changes. You can see that we've in fact been out to these sites in South Carolina and Georgia, making collections. And we've in fact actually lost some collections in from Alabama. So these are plants that actually didn't survive or seeds that didn't germinate. So over time we can track the representations. So we have some new ecological representation here. And our sampling representative score has actually gone up because we have a number of different seeds that germinated from the collections, even in these two sites. So we can see the changes in our collection over time. And we'll continue to use this as we go out and do more collecting as a network, and continue to fill some of these gaps that we now see in Alabama, and parts in Louisiana as well. So before I hand it over to Dan, I just wanted to, to say thank you. And we certainly have our contact info there. We're happy to take questions after Dan is done with his demo. But certainly afterwards as well, please feel free to reach out to us. But I'll hand it over to Dan to show us the live demo.
Thank you for that, Jean. Really awesome to see the live updates coming onto the map of new collections. Exciting stuff. Okay, are we seeing the Gamma application currently?
Yes, we see it perfectly. Yep. Thank you, Dan.
Thank you. All right, so live demo time. Always the most exciting, maybe not always, but can be. Great news about this is we will be sharing the link to the application in its current state with the viewers and participants here today. So you would be able to participate in a live demo yourself on your own time, on your own computer. As it's been referenced so far, we really are aiming to try to make this a user-friendly and approachable tool to support people who may not have a GIS member on their team, or someone who's able to complete the full programmatic R gap analysis methodology. As part of that, we are trying to find or provide some additional input to refresh here. How to run the tool itself, and this will all fill in over time, but we do have a little bit of information here. Again, just for context, we are aiming to kind of have a stable long-term release of this application in early 2027. So what you see today, I think, is a very good representation of what it will look like then. Just hopefully we'll be rounding out some of the edges and getting more feedback and more input from user communities. So let's just go ahead and get started with it. The Gamma tool is set up to allow you to compare your own collection through an upload of data against what we currently have to represent more of a larger communal collection, and that's the information stored about the species on GBIF. This application, just as a whole, does not keep any data. There's no real opportunity to make things persistent. That comes with its pros and cons. The biggest pro is that if you have information that is close to you that you don't want to share with the rest of the world, uploading it to this application will in no way make that publicly available for anyone else to see. The downside with that is that if you are working on your map and you are making edits and you're changing your data sets and you walk away for an hour and come back, the application is going to kick you off and you will not be able to revisit your work in that sense. So We do have an export button here to help you kind of save through file management on your local computer, but it's not something that we can manage, nor do we want to manage your data on our side of things. So let's just start with the upload or custom data. This is probably, I think, in many ways, the most difficult part of utilizing the tool, in that you have to have a very, very specific file format structure, including all of these column names to a T, case sensitive, space sensitive, all of those good things. This can be kind of difficult to get right. So what we've done is provided an example file. So you can download this feature here, and that's going to give you basically a template structure in which you can add in your own data. So I'm just going to go back and browse and re-add the CSV file. Okay, and this does a few things for us right off the bat. It adds points to the map. So we're looking at another magnolia here. You have attribute data on all the points that you can hover over here. And we also have a table format, so you can view some of this in more detail. If you're uploading data, this is very likely your own data, so you should know or should be familiar with the descriptions and meanings of each of these columns at this point. The map itself gives you some functionality for trimming and adjusting your data sets. For example, if you were to select one of these points, it highlights the point on the map with a little blue ring, as well as in the table. And this works in both directions, such that I can unselect points and select from the table to see where they are on the map. If I have a selected feature that I'm not interested in keeping, I can highlight those either through the clicks or this little box to draw an area of interest. Once those are selected, I can delete them with the button down here, and that's going to remove them from the map and the table. You can undo one deletion currently, so use that sparingly, and if you need to make a mistake, it's okay. Just you got one chance to undo it, or you're starting again with your data processing. So again, we should be pretty happy with your upload data coming in, and then the next step is to expand upon it with publicly available data from GBIF. We're using GBIF taxonomy to run these queries, and it should be encompassing of all the plant species that they recognize and maintain in their system. So that turns into a very large set of options since we do have quite a few plants on this planet. And we can query or search through that with some just autocomplete. So if I start looking for magnolia, it'll fill in. I can select. Once you select a genus, it'll give you all the species from that genus. So again, you can either scroll down or just use the autocomplete functionality to find the feature that you want. At this point, you need to select the taxonomic rank. If you pick species, the query ends here. If you pick varietal, you have to select the intra-specific epithet. So we'll stick with species for today. And once selected, a few things are going to happen. It's going to give you a quick summary of the GBIF collection. So at this point in time, we can say that there are 2,800 observations with coordinates. It's recording 9 of what we're calling germplasm or living records. And then just as a summary, there's 2,200 of these are coming from the iNaturalist data collection source. Can tinker with the number of occurrences. It's currently capped to 1,000. We'll keep evaluating how we can bump that up, but I think that's quite good and proper for getting the extent of the species range. And let's go ahead and just download. This taxonomic ID can be quite helpful. We are trying to provide a lot of functionality for engaging with GBIF data, but if you ever want to just go to GBIF itself and use their querying potential, this is the taxonomic ID for the species. Okay, so GBIF has loaded, and we can see that we have a different look on the spread of the species on the map. Much like our uploaded points, you can get a little bit of attribute data associated with each of these features. And then you have a table as well that you can look at. This table you can sort, filter, functions reasonably like an Excel table, other than you can't edit it directly. One thing that catches my eye looking at the distribution is there's a point way out here in Texas, which is quite far from the core area, as well as up here in British Columbia. We could just start to investigate these by selecting them on the map. And we'll check them out on the table. Okay, it just says they're from British Columbia. And the nice part about the GBIF data is that the accession number contains the link to the GBIF accession page. So if you were to select this, you can get to the source information for the collection. So right off the bat, I could see this is University of British Columbia Botanic Garden Collections. Way out there on the peninsula, enjoying life, growing. But I think it's fair to say that this is unlikely to be a wild-grown specimen of the southeastern magnolia. So these are things that I feel pretty comfortable excluding from the gap analysis itself. So I still have those points selected, and I'll just delete them. Go again and delete. So now we're much more centered in Southeast. We have quite a few records to work with. 182 herbarium or reference records from GBIF and our upload records. So let's go ahead and just move over to the gap analysis page. Here we see a similar map, but we lose the source information. So we're just treating these all as the core records for the species. You can turn these on and off. You can change the base map because this map is going to reflect what you produce in your report. So just keep that in mind. For the gap analysis itself, we do provide a couple different buffer distances. Historically, this 50-kilometer has been used for most species, but I think there's a lot of smart and appropriate use of some of these smaller distances in more recent gap analysis publications. But again, it's up to you to decide. That occurred very quickly, but I'll just show again where this current little box is. When you run the gap analysis, you do get a little bit of notation as to what's happening. So too fast to read at the moment. But here we are, we get an output and we get our specific conservation metrics. So we had a good explanation of these already, but you can see them in a bit more context here. The buffer area is effectively how we're defining the expected distribution of the species, where all the area inside of here is included in the species area in which it occupies. We do have some water masks being applied. They don't do great in these inland areas, but they do a pretty good job on the coastline. So this is quite limited to terrestrial species at the moment. And then we have our gap layer. So you can see these nice big holes in the center here are are areas of the distribution that were within 50 kilometers of a germplasm point, and everything else is outside of it. Ecoregion, it's telling us that there are these unique ecoregions that the species was observed or collected within, and then only these 3 have are within the radius of a germplasm collection. So if you were happy with all of this here, you can download your report. This is going to, again, give you a little waiting box. Please be patient and don't hammer this button 8 times in a row. We'll download that, and let's pull it up really quick. And this report gives you, a quick summary with the statistics. So summary, conservation scores, more information, definitions of the features that were used. And it's hopefully portable. It's an HTML file format, so you can share this file over the internet, email, things like that, and you can open it in your web browser. So it's quite flexible, and it gives you this interactive map, which can be, I think, helpful in discussing conservation steps around these species. So more work to do to polish these things up, but this functionality is currently up and running in the version of the application that we'll be sharing. I want to just go back. Let's say you've done your initial assessment and you're really quite unsure about this species out here in Texas. So we can go back and we'll just delete it for our observation. This is going to give you a pop-up saying that you've modified the data set and the previous gap analysis results have been cleared from the map to prevent inaccuracies. So all this is saying is that if you go back to the gap analysis, you will have to rerun it because removing points will inevitably change your conservation score metrics. If this is the kind of core set that you're happy with and you want, you can export the data. This will, well, it's not producing the right object. Okay, bug to note. But this will give you a CSV that combines both of these tables into one feature. So you can engage with it in Excel or whatever spreadsheet software that you like. I did want to take a second to highlight some of the other GBIF filtering functionalities. So if you were really specific about when you wanted to look for a select record, you can change the features here. So we can do a date range. We can potentially exclude any iNaturalist records from your collection. This leaves you with effectively herbarium as the primary source from GBIF. Taxonomic synonyms can be folded in. And then again, this is just relying on GBIF's taxonomy. We can't get a lot of flexibility on that within the application itself. And then the random selection, I think, can be quite important. The way that this filtering process works is that it tends to pull currently just the most recent records. So you can see all these collections are coming 2025, 2026. And then up here, our germplasm records are from later periods in time. So if you do a random selection, rather than returning a date ordered list of observations, it'll give you more spread across time. So yeah, that is the core workings of Gamma. And just to illustrate the point here, if I were to refresh the page, sorry here. This thankfully explains why we were seeing the errors that we were seeing. So totally restarted the application, but all your work from the previous set is gone. There's nothing stored there. So if you do timeout or if you do hit refresh, this page gets reloaded entirely. So I will show just, this is not a very compelling example, but if we were to export this data now, you get a CSV of that input data that's in your downloads folder on your computer there, so you can open that and engage with it in the software of your choice. So okay, I think we will leave it there for the demo. Thank you all for sticking with me through this.
Thank you very much. That was super interesting and I think very applicable to genebanks as we also struggle to rationalize our collections and find out where we need to go and collect. We have a question. Let me read it out to you. Thank you for your presentation. She says, my question relates to using this tool for gap analysis on wild crop relatives. Where the goal is to maximize the conservation of genetic diversity. Does the tool take into consideration the environmental conditions, such as bioclimatic and edaphic variables of the occurrence sites for a given record? This information would be particularly valuable for identifying adaptive traits in crop wild relatives of conservation relevance that have not yet been collected.
Yeah, great question. I would say we do our best to try to proxy that environmental variability through the ecoregion layer. I'll be the first to admit, I think that's a fairly coarse proxy if you're looking for a specific site adaptation of individuals. But that's I think the best option that the current tool provides.
Okay. Yeah, great. I had a question about the data that needs to be inputted before you run. So with genebanks, we have something called multi-crop passport descriptors, right? It is set by FAO. We have several versions of them through time, but that is what genebanks strive to have as passport data, which is very similar to what you have presented here. So accession number, taxon, collection date, longitude, latitude, locality, all of that is collected. There's something called current germplasm type that you have there, which is essential. What is that? Is that like a biological status? Is it a crop wild relative, or is it something else that would need to be created in order to input that? I think it's a requirement to input that one. Is that right, Dan?
Yeah, and I think genebanks have it easy in the sense that we are using that class to differentiate between a living collection and a reference collection. So living collections being the collections managed by genebanks, whereas the reference collections are often herbarium records and ever increasingly the iNaturalist research grade observations of plants in the wild.
Okay, so when you have a 6, I think you have a 6 there. What is that? What does that represent, for example, in that example?
The current we have two options there. So one is a G to represent germplasm. The other is an H, which has historically been referring to herbarium, and we're working on terminology to refer them as reference records. I think because it captures a bit more of the collection variability that's present in that grouping.
Okay, great. So that would be an easy thing for genebanks to create because most of it is germplasm, or not a herbarium in any ways. The other question I had is one thing that we struggle is finding out what is redundant. And all of you mentioned the redundancy. We tend to go to the same place to collect the same materials. So we've ended up with a lot of material in gene banks that is potentially the same, right? So how would this tool fix this problem, help people rationalize their collections, not only within their own collections and the diversity, but also collections, for example, between international centers and national centers who have tended to collect in the same place, and now we have, dots and collection sites that are basically the same. How would that tool help gene banks in the rationalization of their collections?
And Mike, I think we probably all could answer, and so I can first take a stab, and then happy to have anybody else. So I think this is part of where the working with and through this concept of meta collections with other gardens and trying to share data within our part of the GCCs are to really be able to get us to share data with each other. That's part of so that our collections can help us to understand what other collections are out there and who we are working with. Part of Jean's work with the pyramid data has really focused on not all of that material coming to ABG and creating maps that are just ABG collected, but of our partners that are part of a meta collection. And so this is where being able to create data sets with more than just our data is really important. And the tool can take that data if we have these data sharing agreements with each other. And so this is where I think that collective power of working through the networks is so important, because we can all then come to the agreement that if we're all working on pyramid data, we will all share our data so that they know what we have. So our partners, let's say at South Carolina Botanic Garden and the Dawes Arboretum and ABG, all know what material, and we can actually share and input that data. So that will help us to then utilize a larger data set. And so it's really about that data sharing agreements and the networking around, I think, the consortia that are so important. And Jean, Dan, if you want to add anything.
I think for me, I think very much that visualization of where the records are is so much easier than scanning through our spreadsheets of where those collections came from. But to see that, in fact, we might have 100 accessions that are coming from one dot in the map, very much shows how much over collection there could be at a given place, right? And to help us diversify, as you're saying, of where our collections are from, and maybe dispersing those to other places, not going back to that area for a while to let to not be over collecting there.
Yeah. Thank you very much. So anyway, we're
if I could
at
add
the
just
top. Yes, Dan, please
one
go
small
ahead.
point to that.
Yes.
So particularly in the upload data process, I mean, it can accept data from anywhere. So if you're pulling from Genesis, if you're pulling from Green Global, like all those things can get brought together on your end and then put in one map. So you're seeing various collection sources all in one place. And I think that, that's really the best way to tell the story is where do the dots all line up? And, you can try to interpret what the next best step is from there.
Yeah. I think this has been a fascinating talk, and I really want to thank the three of you for putting in the time to teach us, the gene bank people, that there is a new tool out there that we can use. And Dan, the website is available. We'll have a link that we can possibly share on the Grow website in the Crop Trust, and that way people can test their own collections. Is that correct? It is available for usage by the gene bank community, and it is free, right?
Yes, no logins, no personal information collected. Thank goodness, because that'd be a total nightmare to try to maintain all that.
Yes.
But yeah,
So
we'll get the link out there, and just note that it will be changing over time. We're still getting feedback, and then that early 2027 is the hope of something stable and lasting from there.
Wonderful. Well, thank you very much, the three of you, Emily, Jean, and Dan, for a wonderful presentation, especially because it's so applicable to genebanks and I think a very useful tool as we rationalize the collections. Thank you to you, the participants that have logged in, and to Faye in the background, keeping everything running great. Last words and closing this webinar. Emily, please.
Thank you. I just think this is really exciting, and a huge thanks to, I think, the gene bank and crop wild world that really created this concept of gap analyses that we were able to learn from and adapt to conservation. And I think this is so exciting, and we're really thrilled to get feedback and have anyone's engagement and involvement in the future. So don't forget to email us if you have questions, and we look forward to the final product in early 2027. And
Thank you very much. Yes, thank you very much. Goodbye, everybody. See you in the next Grow webinar. Bye-bye.